Right. All right, everyone, we're ready to go ahead and get started. Thanks so much for coming out. I know we had 2 meetings in a row in the Boston side, so next month we'll be back north in Cambridge. So really excited to see the turnout, and really excited to have Isaac Lutz here today. So Isaac is a Baker Lab alumni, and he's been in Boston for a couple years now and been working on a cool stealth startup called Aptamino, trying to engineer orally administered biologics. So yeah, really excited for your talk today, and please take it away.
Cool. Thank you, Chris. All right, so as Chris mentioned, my name is Isaac Lutz. My background is in bioengineering and computer science. So I did my undergrad at MIT in Boston here in course 20, BioE. After that, went on to Dave Baker's lab, as really mentioned, or as Chris mentioned. And so there joined around sort of 2019 to do my PhD before coming back here and actually working at a different biotech startup for a little while before going full-time on actually building Aptamino to build, as Chris mentioned, these orally delivered antibodies. And so the subject of the talk today is sort of looking back at the last 7 years I spent in the field of protein design, the sort of ways that I've seen that we solve problems, where I see the frontier is today, and sort of what we're going towards that I can see rapidly approaching. And so I wanted to frame this as sort of where I see the current frontier of protein design, which is specifically problem formulation. And so I'll get into what that actually means. So the framework I'll use throughout this talk applies to all sort of protein design problems since we started solving protein design problems. So what this actually looks like is relatively simple. It's 4 steps. I'll keep referring to this as a way to sort of think about this. But basically, we formulate the problem. We describe what we actually want to solve with protein design. Then we need to identify a solution space. And so this is the sort of way that we will actually begin solving the problem. And then we sample within that solution space to actually start producing those solutions. And then finally, we evaluate in some form. And so this last step of evaluation is actually what I'll be talking about, at least in this presentation, but still wanted to mention it as an important step here. And so 3 sort of themes that I'd like to discuss in this framework specifically are creativity, diversity, and abstraction. And so these sort of guide us towards where I think the field is headed in protein design. And so creativity, by this, I mean protein design is fundamentally a creative act. And so we need to invent things. We need to go sort of out of distribution to solve protein design problems. Because if we didn't, there would already be a protein that exists to do that. And so this sort of creative act can occur across all steps of this process. But this is what actually takes us from sort of the problem setup to getting to a solution that actually works to solve the problem. And then diversity in this context is how sort of broadly we sample, what sort of solutions we solve, or actually sample within the solution space, and also the ways that we sample these solutions. And so this is what allows us to make sure we're actually finding solutions, depending on how difficult it is to solve the problem. And then finally, abstraction is the big one here. And this is the way that we can actually scale creativity and scale diversity to solve harder and harder protein design problems and move towards problem formulation as sort of the key task of the protein designer to actually do in this rapidly evolving field. And so I'll keep referring to this in the context of a bunch of different protein design problems that I've worked on. And so I've framed this as from parametric design to agents. And so this is starting from when I first entered the field in 2019, where things were pretty manual, pretty sort of basic in terms of what we could do with protein design, all the way to today, where we have these really powerful models, really incredibly powerful intelligence, and we can do far more. And so I'll frame the story through this sort of arc of different projects. And so I won't bother reading all of these right now because there's a lot of them, but we will get to all of them. And so the first one is this project on parametric sampling for heterodimer-based protein logic. And so when I joined David's lab, this work had been produced recently, which is the programmable design of orthogonal protein heterodimers. And basically what the authors on this paper did was create these heterodimers using simple parametric sampling. You install hydrogen bond networks at the interface that confer stability across these heterodimers. And they were able to do this for a whole broad sort of set of heterodimers and show that they interacted orthogonally and specifically, obviously very impressive. But one sort of key shortcoming of these heterodimers is that they required being either co-expressed or denatured and then annealed to actually function as heterodimers and pair appropriately. And so this actually came down to the fact that each half of the heterodimer, so the monomer, was not stable as a protein monomer. And you can kind of tell from the structures because it's sort of just a helical hairpin. There's not really a well-folded core. And so it doesn't behave very well on its own as a monomer, and it actually forms these irreversible homodimers. And hence, that's why you need to denature and reanneal to have this sort of highly specific orthogonal behavior. So this severely limits the downstream sort of applications of these heterodimers. And so the actual problem formulation of and then the identification of the solution space here is to redesign these so that they reversibly form homodimers and therefore can find their heterodimer partners. And the way we do this is through identifying this solution space of adding this third helix to the bundle to buttress it, provide an actual stable protein core, and allow for that exchange. And so this is the overall sort of identification of the solution space in the framework that I mentioned, but then we have to actually sample that helix. And so that's what we're doing on the left. So to sample with that solution space, we need to capture some diversity to make sure we're finding real solutions. This is all done very manually. And so you pick actual parameters that describe these alpha helices. You use that and sort of pick which parameter ranges make sense. You have to find a way to grid sample or randomly sample within those ranges to get sufficient diversity without having some vast amount of scale that becomes computationally intractable. And this is what allows you to actually parametrically sample within the space to find these solutions. And long story short, this works. You add this helix through very manual redesign. What we can do is enable homodimer exchange. And so you can see this is a binding assay where we see the in brighter colors, the heterodimer pairs are able to bind with higher affinity, and the homodimer pairs bind with weaker affinity, and they have the appropriate binding. And key to this is that these were separately expressed, separately purified, and then mixed together. And so that proves that we can actually enable this homodimer exchange through the redesign. Yes.
Oh, super cool. How different are the protein interfaces in this compared to the obligate heterodimer work?
Great question. They're mostly the same. And so part of it was to sort of preserve those interfaces because you have these hydrogen bond networks that at the time were very difficult to design. And so you want to make sure you preserve those and not break them in this redesign process. But there was a little bit of modification there. Cool. And then we were able to solve the crystal structure and show that the structures actually match what we were seeing in the lab to really sort of have the final proof point that this actually worked and this redesign process worked. And then what we were actually able to do downstream of this is enable a protein logic application that was previously impossible, where you can mix these different heterodimers in combinations, have these sort of promiscuously interacting networks of heterodimers, and actually do complex computation and logic gates built out of these subunits. But that is all still unpublished. And yeah, so getting back to the framework here, this was sort of pre any machine learning models, purely just Rosetta, purely parametric sampling. And at this point, there's not really any abstraction at all. And so the creativity comes down to identifying the solution space in a very manual way. So picking, hey, let's put a helix right here, and then actually sampling in that solution space also in a very manual way, picking diversity in a very manual way. So overall, a very sort of tedious process. But to me, as a first-year grad student, this was incredibly empowering because I saw like, hey, we can actually design these things. It's not that hard. You can use a simple math equation to actually design a protein or redesign a protein in this case. So to me, this sort of opened up the box of what was possible in protein design. However, because of these limitations, it sort of felt like what we need to be able to do is rather than manually picking parameters to actually describe the solution space, we need a way to actually sample more automatically. And so this is when I started thinking of sort of more ways that we could do protein design. So this sort of motivates the next method here, which is navigating a native fold space via latent interpolation. And so this was sort of early 2020. The idea here is still sort of pre too much machine learning. And the idea is, can we find a representation of protein folds that allows us to sample with this reduced dimensionality in order to generate novel samples within that fold space? And so how we actually do that is that we fit helices to a given fold space using a helix fitting method. And then that allows us to actually capture something very complex, like a protein fold, with a relatively small number of dimensions. And so in this case, we're fitting ideal straight alpha helices. So very simple. There's only 7 parameters per helix. So it's XYZ translation, phi theta psi rotation, and the length of each helix. And so what that actually yields is for a given 9 helix tet R fold, you get 63 parameters. And then I did this across 236 tet R structures, fitting with these helices. And so what was pretty cool to see then, would not be that surprising now, is that this sort of dimensional reduction and capturing these folds, once you cluster, you can pretty quickly see that there's 2 sort of distinct fold spaces within the set of folds that are captured by these helix parameters, as you can see here. But then I wanted to do more there. I wanted to use this as an actual protein design method. And so we can project these into PCA, or we can do PCA to project these into principal components so we can reduce the dimensionality even further. And as you can see within these 2 principal components, you can sort of see the clustering of different folds. You can see where the densities are that captures the sort of space of these tet R folds. And so then how do we actually sample from this space? So I did something pretty rudimentary here, but basically just sampled a grid of points in that space. And then the way that PCA works, you can just project those points back up to the full 63 dimensions. And there you have it, is your protein fold. And so what that actually looks like if you traverse these points in order is a fun little animation. So you can see from this that basically this parameterization is able to capture where these helices lie relative to one another in a much lower dimensional space. You can see some helices, like the one at the top left in red, have a lot of movement across this fold space. Others don't move so much. And you can also capture how those different secondary structural elements move relative to one another. So super cool, but not really that useful for protein design, because fundamentally this really works just for the solution space of a single native fold. It only works as far as the actual examples of that native fold that we have to sample within, because we're really just interpolating within this reduced space. We're not really going into anything new. And so this, to me, was really like not satisfying, because joining David's lab, I really wanted to do de novo design. I wanted to sample outside of native fold space, really go out of distribution. So this felt too limited in too many capacities. And so going from there, I decided I wanted to do something a little bit broader. I wanted to be able to abstract away for the second and third steps even more, so that we can sort of solve arbitrary protein design problems, more automatically sample, figure out how to do this very sort of tedious manual parametric sampling that I'd done to begin with, but in a way where I could really scale it to solve sort of arbitrary protein design problems. So this brings me to my third project, which was sort of the core project of my PhD, which is developing a top-down design for producing protein architectures using reinforcement learning. So already sort of discussed this, but basically the goal of this method is to tackle those 2 center categories in a way where we can basically just formulate an arbitrary protein design problem in any number of sort of ways, however you would sort of want to capture what you want to solve with the problem. You allow this method to actually find solution spaces on its own, so sort of abstracting that creativity away so it can find its own solutions. And then the sampling is also captured in this process all automatically. And then finally, the evaluation step is actually sort of how you formulate the problem. And so you provide these evaluations that tell the model what you want to actually see in terms of a solution to the protein design problem. And so the actual sort of core building approach to this method is just simple fragment assembly, and in this case of ideal straight alpha helices and short idealized loops. And so what this actually looks like is you start with a stub just in space, in 3D sort of Cartesian space. You extend an alpha helix by some length, you add a loop, you extend another alpha helix, you add a loop, and so on, and pretty soon you built a protein. And so a lot of this was built off of sort of the understanding of what Rosetta was good at the time. And so we could build alpha helical structures really easily. We could do these for a lot of different sort of protein design problems. Anything beyond that was definitely trickier. And another huge limitation of Rosetta and these sort of pre-machine learning models was loop design. And so here we're really removing that as sort of liability by focusing on short idealized loops that we don't really have to design too much, that are already somewhat pre-validated from a library. And so what this process actually looks like to do in a more of a sort of automatic fashion is we can build this into a tree search approach. And so here we do Monte Carlo tree search, where each of these steps is a decision in a tree. So you're deciding between lengths of alpha helices, you're deciding between different loops to append to the structure, and then you sort of search down through this tree, making a decision at each point, at first just a random decision. And this is what actually allows you to automatically create structure. But then key to this is that at each step, we're doing some checks. And so we're checking geometric constraints. So we want to make sure that the structures don't clash with one another. And so we have a function for that. And then we, at the very end, when we get down to sort of like a leaf at the end of a branch, we want to see final evaluation of a set of score functions that describe, did this work well? Were we able to sample protein that we wanted? And so to begin with, we just have sort of core formation scores that describe, do we have a stable protein core? We have monomer designability scores, which are sort of how are the C betas pointing within the core of the protein? Are they designable? Can you actually use those orientations to design a protein core? And then finally, this helix packing metric, which is sort of a callback to the heterodimers, where if we have, we actually need that nice core to the protein. And if you have a helix sort of dangling off, you're not going to get a good protein core. And so we evaluate these at the end. And then the sort of key process here is that as the sort of first initialization is all of these choices is just uniform random probability, but we can take the score functions, have a sort of composite objective function based off of these score functions, and then feed those scores back up the decision tree such that we update each probability at each step so that the next time you go through the tree, if it scored well, you're more likely to pick that sort of route down the tree and sort of bias sampling towards better and better samples. And so this works. And so we, along these scores, we can improve over time. And so here we have those 3 scores that I mentioned, over 10,000 iterations. And so a key part of the development of this approach was that you had to be able to iterate very, very quickly in order to reach thousands of iterations in order to see good improvement. And so it was a lot of sort of like algorithmic development to make sure that scores were computed quickly, that everything was done as sort of lean as possible in terms of computations.
What is your core formation scores?
The designability score is RIF. The core formation is, I haven't thought of this in like 5 years. It's like percent core by sidechain neighbors.
Okay.
Cool. And then what's really nice about this method is that you can actually provide sort of arbitrary constraints, arbitrary objective functions, and you can also do so with completely non-differentiable functions. And so this was actually before the chroma work showing the sort of like shapes that you can make with proteins. We were able to show that you can provide these geometric constraints as purely just hard cutoffs, so not even anything differentiable, and say, hey, sample structures within a sphere, within a cube, within any sort of arbitrary volume, and the model is able to do that and is able to strictly not like violate those constraints. And so you can also do this with more complex surfaces. And so, for example, you can just export like a surface from PyMol. For example, if you want to design a binder, you can start with an initialized point on that surface and then sample structures on that surface as well. And then also this happens really quickly. So this is 50 times a second. You're going through the tree, iterating, updating so that we can actually learn through that tree and produce better and better structures. But where this method really became powerful was going for far more constrained and sort of complicated functions. And so this is where things where, as sort of protein designers, we would really start to struggle more and more to sample plausible backbones, especially if we're doing it manually. And so specifically for symmetric assemblies. And so here the process is very similar. You're extending the helices and loops in a monomer, but then at each step, not only do you look at the monomer, but you also compute all the symmetry mates. So for an icosahedra, for example, there's 59 other subunits. You compute the symmetry transforms of all of those, and then you're able to see where the monomer lies relative to the context of all its neighbors as well in this assembly process. And then where this fits into the broader method is we can just provide additional constraints, additional evaluations to our composite objective function in order to sample more effectively these larger symmetric structures.
So the symmetry step is interesting. What if you run and you crash into another copy? Same thing. Is that step, or is it sort of out of the validation?
Yeah, so we have this interchain clash there. And so that becomes part of the check along the way to make sure that as we build out the monomer, it doesn't clash with itself, but then it also doesn't clash with any of the other subunits in the symmetrical complex. Does that make sense? And so in addition to that, we can also provide different constraints. For example, we can use like shape constraints. We can do simple size limits. So sort of like don't build beyond a certain radius or too small within a certain radius. And then last thing here is that we can also add additional score functions that describe the properties that we want of this final structure. So this includes things like a porosity score I developed to make sure we have a closed surface of the capsid. And then also interchain designability, which is actually the same sort of riff score as the monomer designability to make sure that we have the sort of C beta orientations across chains that are actually designable by, in this case, Rosetta. And so what we can do with this is actually design arbitrary symmetric assemblies. All you need is transformation matrices. And so here we can do C5 through C12 symmetries. We can go larger for tetrahedral, octahedral, icosahedral symmetries. And then we can go even bigger with quasi-symmetric symmetries up to 240 subunits. And it's all sort of just provide the symmetry matrices and then do it. And so what this allows us to do is really have a sort of paradigm shift in the way that we design these assemblies. And so prior to this method, all the ways we design cages, for example, in the King Lab, is using these pre-validated building blocks. And so what that means is you design parametrically something like a trimer, a pentamer, you characterize it in the lab, make sure it's doing the thing you want it to do, and then you take those building blocks and you piece them together into these larger, higher order assemblies to make these larger cage-like structures. And so this is a bottom-up approach. And so now with this new approach, we're actually able to take a top-down approach where upfront we define the target architecture. And so this includes any geometric constraints, any score functions, as I've been saying, and then using RL to actually design these building blocks within all of those constraints. And so we're leaving it up to the model to find sort of any sort of creative solution to this problem. And it just, it will work given whatever we provide for it to sample within. And so what this allows us to do is explore a brand new space of these structures. And so here we have a plot showing largely native structures, native icosahedra. On the x-axis, you have the sequence length, which sort of describes the size of it. And then on the y-axis, you have the porosity calculated with that score I described. And so what you can see is that in general, these native structures have relatively low porosities because often they're viral capsids. You need to package something inside, not have it falling out. And then they also are generally sort of over 150 to 200 amino acids, like they're relatively larger. Also, what we have here is what we had made prior to that was de novo, so all the orange dots, and these are the sorts of things at the top left. So relative to native distributions, you have things that are largely more porous as a result of the building blocks that you need to use to build those structures in that way. And then finally, we have what we can make with this reinforcement learning method, where we can make things that are completely out of distribution, where we have very small sequence length, so it's down to as few as 50 amino acids, just very small subunits, but then they actually have closed surfaces as a result of the way that we're building them, so that you can actually make things that have porosities more like the native distribution of icosahedral capsids. And so we showed that this could actually work and actually worked in the wet lab. And so we tested around 300 of these and actually found structures that did it all in one shot. And so what that means is we're able to design the monomer, have it fold together, have it make all the appropriate interactions with the other subunits all in one go, and then just test it out in the lab and see that it works. And so to do this, we solved two cryo-EM structures actually showing these tiny icosahedra. And so this one is actually the world's smallest. And because we were able to use this method and really go outside of distribution, we can make an icosahedra that's smaller than anything in nature by a good degree. And so this can actually be useful. And so because we're sampling so far out of distribution, we can achieve a surface presentation density that is otherwise not possible. And so what I mean by this is that from a sort of immunological perspective, the sort of orientation of things that you present and the sort of density of how you present them actually affects the downstream signaling, the sort of intensity of the downstream signaling. And so we can have very potent effects by clustering whatever we present on the surface of this very tightly. And so if you compare that to a more traditional structure, in terms of the sort of downstream signaling through the activation of this pathway, we can have a much more potent response by clustering so densely on the surface.
It looks like a hit rate of each design. I assume any didn't make it.
Yeah, so I think it was like 2 out of 300. And this was also, I think some of those were at least one of those was like pre-AlphaFold. So purely like Rosetta metrics. And then the other one, I think we had started doing some AlphaFold filtering of just the monomer. And so I'm sure we could do better with better filtering and better prediction now.
Point out 9, how is to feedback the score from experiment score?
So it'd be arbitrarily.
Repeat the question. It's online.
Yes. So the question would be is how easy would it be to feed back the experimental score back into the process up the tree? The answer is arbitrarily easy, but unfortunately the method relies on like very rapid computation of tons and tons of scores to effectively sort of modify the tree search. And so having sort of like a single data point, like you could affect it, but you would need to have these data points at a much larger scale for it to meaningfully impact the search.
Think about balancing all of these scores together where you don't want them to be adversarial to each other.
Yeah, it's a great question. That was like one of the hardest parts of this method was basically how do we balance these scores? So they were through sigmoid activations is actually how we did it, and then sort of add them all together. But what made it especially difficult is that what we would see in terms of adversarial examples is however you set up the score function, the model finds a way to exploit it. And so if you set up the score function poorly, you get things that are technically good but are like actually bad because of how they're because of what the model can do to sort of cheat the system, which is pretty cool to see. But also, I'd say one limitation of this method because it's too computationally expensive to compute most metrics, and so they have to be really, really rapid score functions, but this leaves them more prone to being exploited by the model.
That like this should that the model estimates like sampling distribution of one particular spot. Like, are you able to adjust the score function so you can see distribution? Yes, or even like in distribution.
Yes. Yeah. So the question was, can you sample more outside of distribution or in different distributions or even within distribution? And yes. And so basically you can just say, if you expand the radius, then you'll get things of larger sequence length. If you change the score function so you favor things that are more porous, you can get more porous structures. And so it's sort of arbitrarily easy to sample in whatever space you would desire.
But like the action about this experiments or like tile this space.
Yeah, so we ran some of those and yeah, you can basically tile the space, but we sort of focused on a very key part of the space just because it was interesting and cool.
I remember the whole pages could kind of be finicky and have stability problems, especially when you did the like spy tag fusion of other components. So how does the stability compare with this kind of tailor-made approach?
Good question. I don't remember exactly. I think so we had 2 examples. I think one was better than the other. So I think one was like pretty stable and relatively well behaved, and then the other one was a little bit less functionalizable. So I'd say mixed bag as far as I remember.
Do you think if you had to like engineer humans from scratch, do you think the ideal organism would only have these hyperstable walls, or do you think that's fundamentally incompatible?
I think it's fundamentally incompatible with. Sorry, the question is if you were to if you were to re-engineer humans completely from scratch, would it be good to have all super hyperstable folds, or is that sort of fundamentally opposed to how life works? I'd say it's fundamentally opposed to how life currently works. So if you preserved any of the systems that we currently have, it'd probably be a problem. But if you made a sort of new type of life where that was okay, where there's sort of different rules, like maybe you don't have these degradation pathways, you don't worry about things forming like amyloids or anything like that, then maybe we'd be okay. But it's an interesting question. Sorry, you said have a better hit rate. What?
Side of distribution. You think you would have had a better history?
Absolutely, yes. And so I think that's, I failed to mention before, that's also a reason why we picked this space. Because as you can imagine, if you're going down this tree of decisions, the more secondary structural elements you have, the less efficient the search is. And so it's actually better at doing these smaller structures. It can more effectively explore that space. And so if we were to go longer, it'd at least be less computationally efficient. And so for context, to sample to get to these structures was 1 million backbones, which was like almost 2 million CPU hours. So we had to do a bunch of distributed computing to actually get to that level of compute to get there. So I would say, yes, we could do it, but it would be a lot more difficult than these ones were to get to. Cool. And so this also translates to in vivo. And so what we showed here is that if you put the influenza HA on the surface and compare it to one of the ones from the King lab in clinical trials, you can actually get more potent vaccination or immune activation using this higher surface presentation density as a result of these out of distribution capsids. And then the last problem that I'll discuss that we apply this method to was solving this highly constrained nanopore closure problem. And so here, this is where the method really had a chance to shine, because basically this problem setup is we have two different rings that we want to combine in order to make a nanopore with a smaller aperture. So we have an outer ring and we have an inner ring, and we need sort of like a coupler between the two. And so it's extremely highly constrained. We have this complicated surface because it's an interface between two proteins that are like sort of too far apart to form an interface, and it has this complex rugged surface. And then it's also symmetric. And so we have to be able to sample something that's symmetrically compatible, but also within a very difficult geometric space. And so this is where the method could really shine, because we're able to set this up as a very easy like protein design problem. Like I think I put this together in like 30 minutes. We were sampling backbones and then they just work. And we can solve cryo-EM structures and show that we've functionally made a nanopore with a smaller aperture as a result of what this method can do. So from this project, we see that we can actually abstract in these sort of two middle sections. And so what we do is we basically provide a way to automate this that allows us to really get creativity and diversity at scale. So rather than having the task as the protein designer be to sort of be like, where do I place these secondary structural elements in order to make this solution to the problem? Instead, we have a formulation for the problem. The model finds a way to identify a solution space, sample within those solution spaces, and do so in a very diverse way, in a very creative way as well, that's well outside of anything that we could sort of, with human abilities, come up with in terms of solutions, particularly for these very constrained problems where actual placement is very difficult.
It's kind of come from a perspective of someone who's kind of used protein design to have more data understand, but I was kind of like interested in like how do you arrive on that like problem solving framework or like kind of like inspired it or like
Gotcha. Yeah, like the overall approach.
yeah, the overall approach.
Yeah, so I would say it was largely motivated by sort of just having a very generalizable way to solve arbitrary protein design problems. It was motivated by working off of very idealized elements in order to have things that were very compatible with Rosetta at the time to actually design, so that we could have a success rate that would actually work experimentally. And so these were a lot of the motivations for it. I'd say like this model, as a result of the limitations that were built into the formulation of this method, is definitely sort of aging, I would say. And so like what it works well still sort of beyond other more modern approaches is, for example, is these highly constrained problems, these non-differentiable score functions, things like these symmetries. That's where I don't really see any other models that are able or methods that are able to do this so effectively. But for everything else, it's not very efficient. The actual solutions you get are of lower quality, I would say, and require sort of a lot of design. But I think there's still ways to really make use of it, especially in conjunction with other methods to do more with this sort of approach. Cool. And so I'd say one other thing to highlight here that this is actually a good question to lead into that is that there is, although we're getting a lot of creativity at scale and diversity at scale, what we're fundamentally missing here is a sort of methodological diversity in the fact that we're really limited to sampling these very idealized alpha helical structures with the short loops. And so if you said, hey, solve something with beta sheets, can't do it. If you said like solve certain other types of challenges where you need structures that are more complex or need loops of a certain orientation or something like that, it can't necessarily do that. And so we've sort of imposed a methodological constraint that fundamentally reduces the diversity we can get. So even though we've abstracted, we're still limiting the space of protein design. And so this sort of leads into the next project, which is helical peptide recognition across the protein design toolkit. And so really what we're going to see through this project is that with methodological diversity, you can actually solve harder problems and different problems. And there's a lot to be gained by having a lot of different tools in the toolkit to do protein design with. And so at the time, helical peptides were definitely a new challenge for binder design. So when I started on this project, we had many protein binders made with Rosetta at a success rate of like 1 in 1,000, 1 in 10,000. And they were really sort of built upon these very idealized scaffolds, alpha helical, obviously, and basically just 3 helix bundles that you make into many binders. And so the space of targets that you can go after with these many binders is fundamentally limited by the scaffold itself. So what I mean by that is that because you have something that's very just sort of globular and convex, you can really just go after things that have a nice concave rigid binding pocket where you can fit well inside of that structure and make good interactions. And so you can see this across the targets that people chose, the targets that people had success with. And when you look at something like helical peptides in contrast to that, it's completely different. And so these peptides are sort of wiggly. They actually have a high degree of disorder in many cases, including completely disordered regions. They are entirely convex, and so there's not really a binding pocket to fit within or any sort of convex or concave surface where you can fit a nice mini protein inside. And so the first sort of naive approach to this, which is where I guess the human creativity comes in, is that the helical peptide is sort of like a hot dog. And so the best way to bind to a hot dog is like a hot dog bun, because you can really cradle it, you can make a lot of nice interactions. And so what I did to do this is sample a bunch of hot dog buns. So these groove scaffolds, where we basically have a set of helices that make a nice groove to make favorable interactions with the helical peptide. And so I sampled 18,000 of these, designed a bunch of binders. And then actually what was shocking is that at a pretty high success rate, we could get highly specific binders to these peptides. So I basically tested a 96-well plate against like 7 or 8 different targets, had binders to most of them that were showing good on-target specificity with very little off-target interaction. And this was at high success rate and also pre-AlphaFold. But I'd say one limitation of these right out of the box is that they were not very high affinity. And so these were sort of like low micromolar affinity at best, I would say. And so I also took a stab at this with the Monte Carlo tree search approach. And so I would say this is an application where it worked decently well, but not as well as other methods. And so what was cool about this is that with that tree search, you could find creative new solutions. So right out of the gate, we saw solutions that looked a lot like those grooves that I'd already designed, which was sort of validating and cool. But then it also found sort of more creative ways to solve the same problem and make those sort of interactions with peptide. But then to get to the sort of methodological diversity, this ended up being a much larger team project as we had an explosion of new machine learning methods. We had a lot of new tools come out, and this became a sort of toy problem for a bunch of different people to say, hey, what if we try this new method on helical peptide design now that it's finally starting to work? And so we did a bunch of different methods to go after the same problem. So I'll go through these pretty quickly. You may have heard of a bunch of these. But basically, we started with in-painting these parametric groove scaffolds. So basically in-painting to extend the interface, and that yielded low nanomolar binders. We used pre-validated helical repeat scaffolds that made the same sort of grooves, but had actually already been tested out in the wet lab. Also yielded low nanomolar binders. Hallucination approaches where you provide just a sequence. And then what's interesting here is that the design looks somewhat similar to sort of groove, although it's mostly sort of a longer mini binder, I would say. Here you get sort of double-digit nanomolar binder. And then finally, this is when RF diffusion just came out. And so the next approach is partial diffusion. So taking these parametric scaffolds, adding noise, and then denoising. And there you can get picomolar binders. And these are still the groove shape. And then finally, full diffusion, where you start from purely a cloud of Gaussian noise, diffuse, and get to, another groove scaffold with picomolar binding.
So, looks like they're saying what on the part of the head, say former approaches. See.
Yeah, great question. Yeah, so the question is pros and cons if partial diffusion and full diffusion works, like what's sort of the best method to use here? And I would say largely my takeaway from this is probably just full diffusion because it's kind of the easiest one to get picomolar binders with. You don't actually have to specify anything. And so to me, this is a way that we sort of abstract away from the sort of steps that are necessary to design a solution to the problem and this sort of allows scaling to more solutions, I would say. But largely depends on the problem, largely depends on the method. And so things like Bindcraft, for example, hallucination approach, also sort of hands off. And I would say that is also a very good sort of binder design method. But I think the one in this case was an earlier version of those sorts of hallucination methods that was not so, I'd say, like sophisticated.
Well, you ended up with helical bundles also. Was that strength or did that appear just from the diffusion?
So that just appeared, yeah.
Just kind of eyeballing your structures on the screen there. It looks like your picomolar binders have potentially greater interaction surface area. Do you see a direct correlation between just interface SASA and binding affinity?
Absolutely, yeah. And so that's a great point. And so that's why I would say these in-painting structures worked because they're fundamentally just taking the parametric ones that were weak, extending it a little bit more to make more favorable interactions, and you take a micromolar binder and turn it into a low nanomolar binder.
I have a similar question for live physics.
Mm-hmm.
Showed a variety of different peptides in a chemical.
Mm-hmm.
Presumably in the solution phase, there's a mixture of red, oil, helix, and you look at the CD spectrum. So my question is, are the ones that are more helical in solution because there's less of an project cost entering the hot dog one?
Mm-hmm.
So are those the ones that have a higher, stronger affinity, or is it the ones that have a larger?
Yeah, great question. So yeah, the question is basically like, are things that are more disordered, do they have lower affinity? Things that are sort of more, have more flexibility in solution. Trying to remember, I think it was not a clear answer. It wasn't sort of systematic enough to get a very clean answer there. But I would say maybe there's new work out of the Baker lab, like from the last couple of years, where it basically seems like they've solved fully disordered peptide binding, and I think with very high affinity. So I would say it's not necessarily a limitation from what I've seen, but a little hard to extrapolate from this work specifically. Cool. Yeah, so to summarize, methodological diversity allows us to abstract in a sort of new way. And so here the sort of abstracting comes from just more people. So we have different people, different ideas, different methods. This allows us to identify a bunch of new solution spaces, a lot of them sort of conveniently overlapping, which is cool to see. Also allows us to sample a lot more effectively within these solution spaces, as we saw, sort of diffusion methods were able to do this very effectively and at a high success rate. And so that definitely has value here in terms of a level of abstraction that we can use to solve harder problems more effectively. And so finally, getting into the last story here, this is agentic design of helical antibodies for oral delivery. And this is what we're working on at Optimino. And so what we're doing here is we are solving the problem of antibody instability in order to design oral antibodies. And so the way that we do this is that we structurally stabilize them so that they can survive throughout the GI tract. And so as a lot of you probably know, antibody drugs are extremely effective therapies for a lot of different diseases. They're highly specific because they have large interfaces, and they've really sort of transformed the therapeutics landscape since they were first sort of turned into drugs. But they face a fundamental limitation in that they all must be either injected or infused. And so obviously downsides for patients in terms of compliance, convenience, a lot of other sort of shortcomings in terms of things like cold chain because you need refrigeration for these drugs. And so what we're doing as a company is we are turning these injected and infused drugs into pill form oral antibodies. And so the way that we actually do this by structurally stabilizing them is in broad strokes, we look at the region of the antibody that's fundamentally a liability. So what happens if you were to eat a traditional antibody, it's exposed to the extremely harsh environment of the GI tract, which is made to break down proteins. And as antibodies are proteins, they get digested pretty quickly. It's very low pH in the stomach. They start to denature. And then what happens here is that specifically near the loops in particular, you see this sort of denaturation at low pH. And then this exposes sites of protease cleavage. They get chopped up into little useless bits, no longer work. And so what we do here is we remove those loops, we replace them with alpha helices, and then we redesign the whole thing. And in the process, we can also design a new binding interface using those alpha helices in much the same way you design a mini binder. And then we finally use prediction and selection tools to actually pick which ones we want to characterize in the wet lab. And so long story short, this works. And so what we've been able to achieve is the highest antibody stability ever. And so this is across a number of different sort of assays. And so first in vitro, testing the actual GI stability of these antibodies. And so we test these in simulated intestinal fluids, simulated gastric fluids. And so these are basically gastric fluid, for example, is pH 2, high concentrations of pepsin. And so most proteins just get shredded in these conditions within minutes. And so what we've shown is that we have designed antibodies that can survive for over 24 hours on both of these fluids in comparison to the best attempts to date, which is up to 6 hours in SIF alone. We've shown that this translates to in vivo as well. And so if you feed these antibodies to a mouse and then detect them in the fecal matter, we've shown that we can find the antibody in abundance. And compared to the next best attempt, we're showing over 10 times survival of the antibody throughout the GI tract. And then this sort of translates to thermostability as well. Because they're so stable, you can actually boil these antibodies and they don't denature, which previously was something we only saw with mini binders.
So is this a full tetrameric, like 150 kilodalton antibody, or is this just the
Yes, so this is just the single domain antibody actually. So it's a single domain antibody, aka nanobody or VHH. So starting with a smaller structure, but we've also started applying this to different formats. Larger proteins, and we've seen that it will translate to larger structures as well as a way to structurally stabilize the protein.
Distribution out of the GI tract a problem, or are you just focused on applications within this?
Great question. So starting with the GI tract, but we do have a way to get beyond the GI tract, and so that's also something we're doing with the company, but something I'm not discussing at least in this presentation.
The nanobody is like 7-8 loops. What? How many are you replacing?
So in most of the cases we've tested, we're replacing 2 of them.
So the CDRs remain the same.
So we can do both. We can replace either the CDRs or we can replace the framework loops. And so that you may be wondering about sort of binding activity that you get with this. But because we are replacing loops with these alpha helices, we can design them to bind instead. And so in one single step, we can both stabilize and design for binding.
Why do you focus on the nanobody scaffold then?
Lots of reasons for that. For one, it's a sort of simpler place to start. It's less computationally expensive, requires less helical replacements than a larger structure, and it's sort of the ideal format in terms of balancing this like the stability that we can get sort of intrinsically, that we can design easily, but then also rely on the sort of proven out native scaffold of a single domain antibody.
What we can do is a mouse. You call them biologics. So they bind a target at one time. They will never be degraded by a cell, by whatever. What matters can they be toxic?
So for these gut restricted ones, so for ones that don't make it systemically, they get cleared because they eventually fall off their target and then they leave the other end. And then for systemically available ones, the way that we accomplish half-life extension is with albumin binding instead of a conventional FC receptor. And so eventually they fall off of albumin and then they get cleared by the kidneys.
And you don't see any toxic effect so far?
So far. Yeah, so we've done just one mouse study so far, haven't seen any toxic effects, but obviously it's things that we're testing very shortly in the future. Yeah, it's a great question. And so what we're able to do with this helical approach is sort of tackle immunogenicity from two angles. So the first one, so to the sort of immunogenicity that we would be most concerned with here is anti-drug antibodies, which would basically render it ineffective by just sort of suppressing it. And so to get that, you actually need class-switched IgGs, which involve both B cell recognition of the full structure as well as T cell recognition of the chopped up and presented peptides of the structure. And so we can go after this. Most sort of biologics go after this from the one angle of avoiding B cell recognition, but we can actually go after this from both angles. So we can use these human frameworks. So the vast majority of the surface resembles a human antibody framework. But then also because they're so stable, they're less likely to get chopped up and presented in the first place. So we can sort of have a two-pronged approach to avoiding the immunogenicity.
The sequence outside of the design make it resistant.
Great question. A decent amount that I won't exactly quantify, but yeah, you do have to design a little bit more. Yep.
Are there character confirmations that you can access by using these helices, like maybe concave, convex, sort of thing?
Yeah, it's a great question. And so I think generally we are not too limited in what we can go after, but I'd say there's sort of edge cases. So for example, like if you wanted like a deep sort of binding pocket where you would really need like an extended loop to actually reach inside and make that interaction, that's something that's not really addressable by this. But then that's sort of a situation where you'd also have a lot more challenges with the stability of the molecule if you have such a sort of extended loop interaction.
Does that mean you have to touch the business end? Like you said earlier, maybe you can do the framework only. So I took that to mean you can fix this thing just by putting the helices at the opposite end.
Mm-hmm.
But what you just said, like maybe not.
So yeah, so there is some complexity, depends on the CDR loops, but in general you can. And what this allows you to do is also multifunctionalize the surface, and so you can actually have like bispecific molecules on a single domain antibody scaffold.
No question about like what if the work because all given other everything beta sheet just redesigned on every molecule. So that was the thing other.
Yeah, so we have tested other things. This does largely come down to the sort of intrinsic stability of the alpha helices. And so this is what allows us to actually confer the stability, is that this secondary structural element is so stable and so sort of ideal on its own that we can use that to redesign, and other methods don't work in terms of like trying different loops or trying sort of like all beta sheets as far as we've seen.
Where does the agent
Soon. We're almost there. Cool, let's get there. So finally, now that we've done this, we want to keep abstract. So we have all these different new protein design tools. We want to go bigger. We want to make sure that we are like truly pushing the field forward towards the frontier. And so what that takes is agents, as you mentioned. So basically, how do we keep abstracting? How do we keep taking a step back here and elevating the protein designer to where we can solve bigger and bigger problems in more and more of an automated fashion? And the answer is agents. And so we have to actually connect the wires. And so what we really need here to make this work is number one, we need the agents. They have to be good enough. They have to actually be able to reason over all parts of this problem effectively to do this. And then secondly, we need a map. And this one's a little bit less intuitive, but basically we need a way to connect all these disparate ways, methodological ways, sort of structural solutions, all these disparate ways that agents could come up with to solve a protein design problem. So we need a sort of connective tissue to connect all these points in space and allow the agents to more effectively reason over everything. And so we can actually use protein language models in the latent space from those models as the map. And so the way these language models work is that you feed in a ton of sequences, and then they go through a bunch of neural nets, basically. And then at some point in the middle, you have a sort of representation of the sequence and also implicitly the structure that's captured by a matrix. And so that's in the center. And basically what the information contained within this latent space covers both jointly the sequence and the structure of the protein. It has relational information in terms of where it would be next to other proteins, and those distances mean something. And then also, there's also a sort of implicit encoding of function as well here, because you have sequence and structure, which we know confers function. And so we use this as the map. Go ahead. In this case, we are using, so there's ESM, and then there's this method called cheap from Genentech, where you have these, this is way in the weeds, but basically you have these like really massive activations in ESM that makes the sort of latent space not very smooth. And so they built this method where you can smooth out latent space to make it more workable. Cool. So you may be reminded of the very early project back in 2020. And so this is very similar to this, but now we actually have sophisticated tools to do this and do it much more effectively. And so to start with a sort of toy problem using this map, if we have protein 1 and protein 2, basically what we wanted to do here is take attributes and functions of protein 1 and then transfer them onto the fold of protein 2. And so if you do this in this sort of latent space, you can draw a line between the 2 points, and you can sort of naively pick points along the line. And if you do this, you see that, like, for example, the PLDTT goes down pretty atrociously. It becomes like less viable of a structure, sort of obviously, because why would a midpoint between these 2 folds be a viable structure? But what we're able to do is provide a bias embedding that sort of refines the space. And what I mean by this is that there's no understanding of which parts of each of these structures that we want to preserve in the line interpolated between the 2 structures. And so we can provide a very naive guess at that simply by providing a sequence that contains the parts of protein 1 that we want, as well as the parts of protein 2 that we want, that sort of gives us a random reference point that's a vague guess at how we're going to do this. You can use this bias embedding to create this sort of more curved interpolation. So you're still on this plane in a very high dimensional space between all these 3 points, but we can sample along this curve rather than along this straight line. And so what you see there is actually you start to get further and further away from the 2 original proteins and still preserving SOAR.
You have to do that moving some direction in embedding space. How are you getting the sequence back out at that point?
Yeah, so with this method, you can take embedding and then go from embedding to sequence pretty. Yes, I believe. Yeah, you take through and then you can output a sequence, and you actually output a structure as well. And so we can do this at more scale. And so if you take that whole plane and sort of sample over the whole plane, you start to get a landscape of what things would look like in this plane and embedding space between these 3 different sequences. 2 of them real, 1 of them sort of a bad guess at it. But all of these are still bad, all of these structures, which shouldn't be a surprise because we're basically operating on a extremely constrained plane within dimensionality of over 1,000. So we're missing out on like over 1,000 dimensions here in terms of where we could possibly search within this map. And so how do we search that higher dimensional space? For this toy problem, there's a lot of different methods that you could probably think of, but here we use particle swarm optimization because it's fun and cool. And so the way that this works is we take that plane and we initialize a set of points randomly on the plane as the sort of starting points for the optimization. And then what we can do is actually, the way that this algorithm works is that each point has a velocity in this high dimensional space, so it can go off the plane. And then it's initialized with a position and a velocity. The points sort of move around, and they optimize based off of the scores that they get. But then you also have this global optimization where there's a sort of swarm intelligence where like better scores of the swarm can steer other particles towards that one. And so what this looks like is you can basically watch this swarm of points move across this latent space and discover better and better regions. In this case, we've used a more complicated score function that captures the PLDDT as well as the interface sequence ID that we want to preserve function, as well as the sequence of the second protein that we want to morph the fold onto. And so I think something that's a little bit less intuitive about this is that even though these all look to be on the same plane, they're actually going off into other dimensions. And so it's hard for us as humans to sort of conceive of what's going on here, but we're exploring on a much greater dimensionality that's not quite captured by this representation. And so this swarm is able to sort of navigate this space as a collective. So that's what that looks like. And long story short, this actually works. And so we were able to experimentally validate that we can transfer the function from one fold onto another fold using this approach where we use these particles moving through latent space. And so this basically proved out to me, we did this sort of last fall into early this year, this proved out to me that this map is an effective way to reason over proteins. And we can actually use this map for protein design. Which function? Yes, so the function is binding, and the folds are pretty different in terms of like number of secondary structural elements and sort of arrangement of things, but that's about all I can say.
When you say new fold, do you mean like because there's no other solution out there in nature that did it that way, or just different from their starting point?
Different from starting point, and it doesn't exist in nature. It's like an out of distribution fold, if that makes sense. So it's not something that's within the training data of the language model, if that makes sense. And so now we have the map, and now we can talk about finally the agents. And so to get to the agents, this was really inspired by about a month ago, Andre Karpathy came out with auto research basically showing that an agent can reason over an LLM to actually train the LLM and do ML research on the LLM by itself, and do it basically as effectively as human researchers. And to me, this was a sign that these agents are ready to do protein design. And so the question is, can agents reason over protein design problems? Let's find out. And so the toy problem here is optimize a binder by reasoning over protein design tool parameters and sequence as well. The sort of baseline measurements that we're doing here, so we have 10 agents doing this. We also have baseline 10 GPUs with just sort of a naive random parameter settings and no control over sequence. So this is something that you would do as sort of like a production run already, where you just say submit a bunch of jobs and then filter down to try and find things that work, but you don't have an understanding of what parameters are going to work without a bunch of human hours to sort of iterate and figure out that. And then this is where the last part comes in, which is this is actually a problem that I did the week before. I spent several hours of my time on it, many more computational resources, in this case, hundreds of GPUs. And I reasoned over sequence selection. So actually like the choice of sequences to feed back in rather than individual amino acids, because I do not have the patience for that. And so that's sort of the level of sequence reasoning that I could provide to this protein design problem feasibly within my time. And so what we find is that they're very good at this with this limited information. And so if you look at the baseline, this is just, the score doesn't really matter here. This was true across a lot of different scores, but here it's IPTM. You have the baseline mean of, if you just have the 10 GPUs running through many, many steps, the average obviously doesn't go up because you're sort of randomly sampling, it just jumps around. But then the one we care about is the sort of best, so the cream of the crop that we're selecting that we would actually test out in the wet lab. And you can see that with the baseline, it sort of goes up and then it plateaus because we get diminishing returns as we scale computation. However, if we use the agents, the mean goes up pretty much immediately because they right away start reasoning over the sequence, identify things that we hadn't already. They start tinkering with parameters. And in not many steps, they're able to approach the best that I was able to obtain over hours in a large computational cost. And so I sort of ran some number estimates on this, and it's actually more cost effective to do this and have the agents reason over these things than having myself do it, or the sort of naive just like running too many GPUs for much longer.
You say agents, are you provide more details, like how exactly are you like which agents and what models?
Yep. Yeah, so I started with like pretty weak agents that could fit locally on the GPU. They weren't very good. They actually did work. They improved a little bit, but then I upgraded to Claude through the Claude API and a sort of programmatic loop, and it sort of blew everything out of the water.
You gave Claude like Claude code, and you give it access to all these tools on your local machine. Is that
Yes. So it's the API. So it's just like a non-interactive bash call functionally, but it has access to all the tools and then just uses them, writes its own scripts, does whatever it wants to do in between each iteration, and then it's evaluated. And then there's like logs written at every step.
Experimentally test any of the binders?
We're testing some tomorrow. Yeah. And so what really stood out to me is actually I fact checked the model. Obviously, I didn't want to see hallucinations. And this is what like truly sold me on the model's capabilities. And so I checked all of these with the structures like pretty thoroughly because I wanted to make sure it wasn't hallucinating. And you see pretty crazy domain expertise. You see pretty crazy reasoning at a scale that I would not be willing to do over thousands and thousands of structures. So for example, it is able to reason over chemistry, protein biophysics. It also is able to implicitly reason over sort of fitness landscapes because it does things like it finds one sort of place where it's fully exploited and is not able to improve that sequence at all anymore. So it goes to the second best sequence and then takes that as a new input to try and get a better global maximum, which is pretty impressive. Also, all of this it does purely over sequence, but it's able to infer the structure as well, purely based off of sequence alignment, the history of sequences. It can say, oh, I know this one's interface and it's correct. It knows like this one's on the surface and it's surface exposed, and it does this correctly. And then lastly, it does things that you don't even ask it to do, like do a consensus sequence to feed back into the model as just ways that it's sort of creatively coming up with new solutions to the problem.
The target that's well understood. Sure. Like how to bind to it, but it's like
It's new.
But the target structure, is it hyperfolded or?
In this case, it is from the PDB. Yep. But it's not reasoning over that structure at all.
Interactions. It's reasoning over interactions, right?
It is reasoning over the interactions. Yep. But interactions that are not present in the PDB.
Yes, but it's
Yeah.
Binding site is active between the brackets then not already.
Yep.
as you're looking through the logs, you see things that it mentions that are incorrect return. Like looks like the partial T is probably like Susanna's paper on the partial diffusion of the binary space.
Mm-hmm.
I guess I'm wondering, like, did it ever come up with strategies that are current? I mean, I'm guessing Claude's seen all these papers that have been published in beauty, and then implemented them for you, so you don't have to. But like, does it go beyond that, or?
So far, it seems like it does, or it at least does things that are new. I'll get into that a little bit later, but yeah, it definitely does some of that. It definitely at least adapts things in ways that it wouldn't do previously, or that are not like in the literature. And then one additional thing I'll say there is like some of the like individual amino acids it's reasoning over in interactions are like they're purely on just like a sequence level. It doesn't have any sort of literature to refer to, and so it's basically coming up with a strategy just based off of like vague biophysical, like biochemical understanding, like alignments of sequences, and sort of what it can reason over in terms of those things that we supply to it.
Got it. So it could act directly on the sequence level, not just necessarily running tools. That you're saying? It propose a hey, fix this amino acid this way.
Mm-hmm. Yes, so it does things like that. It says fix this amino acid. It says preserve these two, design the rest, and a lot more sort of complicated things that I gave it access to be able to do in terms of the models and sequence sort of reasoning that it's able to do. And so now that was just a proof of concept. And so now we can connect all the wires. So we have the agents. They're very good. They're very intelligent. And then we also have the map that allows us to connect all these pieces together. And so now what we do is we pull everything together, have the agents reason over everything. And so what we're doing now is full stack agentic protein design. And so what we have, the model has access to are these protein design tools. It has access to sequence, and it also has access to structure. So you can actually give it PyMol, and it can reason over the structure as well, in addition to the sequence. You have this latent space map that allows it to draw connections between different things and different data points. It also has quantitative and qualitative experimental data. And so you can feed in just sort of natural language experimental data and say like, oh, this protein maybe aggregated a little bit, or hey, maybe this one had better, like you can even do relational data. So you can say like this protein had better expression than that protein in this experiment. And all of this is information that it's able to reason over that you can provide with any amount of structure. And then finally, key to this is that you can also provide natural language context and description of the task. And you can say vaguely, I want to make this a better binder. I want to make this a better antibody. Be more specific if you want, but you don't have to. And you can provide that to actually set up the problem for the agent. And so sort of question there that you might guess is like, is it able to handle all of these things? And the answer is absolutely. And so the way that this works is we have this latent space map where on the map we have experimental data points that we've already tested. And so each of these is in the latent space. It has a description of what the sort of experimental properties measured were, both quantitative and qualitative. And that's sort of labeled data within this embedding space. And here we're using like UMAP dimensional reduction just because it looks a little better. And so what we're able to do is initialize the agents within that space and then have them use all the tools at their disposal to design better proteins. And so they do just that. And so basically over time, you can't really see the lines connecting these, but with each trajectory, they independently start to explore this space relative to all the experimental data, also relative to the points they previously explored. And they're able to do so with whatever tools they want to in whatever way they would like. And so you can see here, the scores here, green is good, red is bad. And you can see that the landscape it starts to explore and covers things that are like you see more peaks, you see more sort of valleys between data points, and the agents are all sort of exploring this overall landscape within the latent space to do design using all the tools that are available to them.
UMAP of the 3 to 14, or what's the UMAP space?
The UMAP is of the embedding space, of the latent space. So rather than doing like the PCA that we were doing before, it's just UMAP dimensional reduction of the 1,000+ dimension embedding matrix.
And the color for like good bad is how exactly is that being calculated?
So for here, this one, I believe this one is just like PLDTT. And it's at a very tight range. So all these are actually pretty good. But what I will say about this that I think is especially interesting is that all of these started at as we sort of know, like the scores break down past a certain threshold. They're not so informative over if something's good or bad. So all these are already starting at good scores. And so it's sort of on the agents here to actually figure out the evaluation part for themselves as well. Because the task is just create a better binder. And so they know that if they drop below a score, it's going to be bad. But then they have to reason over how they can sort of come up with their own evaluations for how will these points behave relative to actual experimental data that I can reason over. Like what parts of those structure do I think are important in order to confer additional properties that I want to grab from somewhere else? They can reason over all of this and come up with their own way to sort of characterize if they're doing a good job or not. And so this is where the sort of like methodological diversity I was talking about comes in, where ideally we would also have evaluation methodological diversity, because if we're all just doing the single same AlphaFold scores, then we're sort of missing out on what we could explore more reasonably with these models.
Data do you buy?
Yeah, so for this example, it's things like binding affinity, expression, like GI stability. It's basically anything that we have and would characterize in the wet lab, you can provide into the to the model and have it sort of identify which things it wants to try and optimize, and then go towards those different properties.
very much faster for
So these are all independent jobs. So these are just run on like T4 GPUs, and sort of independently. And so yeah, so you do have to run it on the cloud, unless you have like a bunch of GPUs sitting around. And so yeah, but because the API call is off machine, so like the Claude call is going to a different is through their API, and so it's on a different machine running the actual LLM, if that makes sense.
if you have something like all it takes maybe like take like a few days to have just one equation. You repeat it over with the equation, it takes a moment. Depends on the tool, maybe.
Right. Yeah, that's a good point. So I think key to this is you do want to have that loop that's actually able to evaluate and provide feedback more quickly to the model. And so if you have something that has a much longer sort of evaluation time, then you you definitely slow down the ability of the model to explore. Yeah, and so what we can actually see in the logs of these agents is that they're able to reason over the full stack simultaneously. So you see things like we're doing latent space interpolation from one experimental design to another. We're finding appropriate mutations that are yielded by that interpolation. We make sure that they're far away from the interface using PyMol, so reasoning over structure. We take parameters explored and sort of tested through previous runs, so the history of hypotheses in terms of sort of an understanding of how the protein design models work. We feed that back in. And then also we we take things from the experimental data, in this case, qualitative experimental data, to understand like what we actually want to do here is the agent has chosen to preserve affinity while trying to improve the GI stability of the molecule. And so the agent is able to simultaneously reason over all of this, decide what it wants to do, create a hypothesis, test it out, and then iterate. And I think key to this also is that the level of knowledge that I've seen reading the logs is like definitely surpasses mine in terms of what they can call upon in terms of reasoning over this. They also do so tirelessly, obviously, so you can run them nonstop overnight. You can also scale, so like we could boot up 1,000 of these today to run and just do this in parallel at massive scales.
So what are the first principles ways of improving GI stability? I'm not aware of any literature that says this is exactly how you improve GI stability. How does the LLM know how to do that?
It has to figure it out. So that's where the sort of scaling the creativity comes from. And so I think like what I have been convinced of by the LLMs and convinced of by like working with various models is that like they can work out of distribution. They can come up with things that are based off of literature, based off of sort of fundamental understandings, but take that extra step and go outside of distribution. This is things that the models are able to reason over and come up with themselves.
You deal with local minima problem, like, are they defining the best solution within one, or how are you forcing it to find all the solutions across that? Because some of these look like they might optimize towards a certain stability, and how do you know, is this a representation of multiple local minimas or a global, what the agent believes is the global minimum?
Gotcha. Yeah, so what I've found actually through testing this out is they do tend to they both explore and exploit, and they do so like with their own judgment. And so basically they will like sometimes they'll find one sort of patch and they'll say like I'm gonna really explore this, like see what I can sample within that patch. And other times they get to the patch, they see scores not necessarily improving or not like not getting the results they want, and they decide to jump to something else and try something else. And actually,
do you see it in that context? Do you see a different set of rules emerging in that second local minimum? Like if you had, just oversimplifying, if you had a beta sheet solution and alpha helical solution, your rules should be very different on how you get to the local minimums of each of them.
Right. It's a great question. And from what I've seen, I would assume that the model can infer that because it has access to the structure and can say, oh, we're working with like a very different structure here. And so like we should reason over this differently.
Had 10 agents that started on the left side.
Yep.
So if you see it somewhere else, you convert to different results.
Yes, definitely. And so I think like key to this is always the understanding that like the solution space and like the space that you can design within is like extremely vast. And so we like if you see even if you see in the same place, you'll get like dramatically different solutions. And so it's basically a way to it doesn't really matter where you start. Well, it matters a little bit, but you want to start with something good, or in this case, you do. But basically, you allow it to explore and then find new solutions and test new hypotheses. But because the space is so vast, it's sort of up to the agent to figure out how it wants to do that.
I think they don't have this data yet, but is there like any sort of computational evidence provide that like there is some signal with respect to like binding any stability or like any of them like beyond like just like taking what the agents say and stuff.
Yep. Yeah. So what I can say is like if you fact check them, if you go through the like reasoning behind the mutations they're making, if you also look at the scores, like they do very much improve the scores more than I can do as like a protein designer doing things manually.
They're like the alpha metrics or
yeah, like alpha fold metrics. Yeah, or some of the scores here. And then also if you fact check like their reasoning over the types of changes they make, or like even like the single amino acid changes that they're making, they do check out. Like there's good sound sort of like biochemical reasoning behind what they're doing. And so to me, it really does feel like in a weird way, it feels like getting back to like far more manual protein design, where you can have an actual like old structural biologist looking at the structure and saying like, oh, if you change this amino acid to that, it would do better or do XYZ. And that actually works. People have done that for a long time, far before protein design. And the agents are now able to do that at a ridiculous scale and with far more knowledge than any given sort of structural biologist. Yeah, so that's a good question. And so, yes, as I sort of alluded to, these scores definitely like they're only useful to a certain extent. And so here, I think this is where we really do start to break down the computational metric evaluation layer, because we want to get to a point where the agents are reasoning over things without metrics, with sort of their own understanding of what's going to be good or what's going to be bad. Maybe that they base that off of their experience, or maybe that they base that off of their collective experience. But most importantly, they base that in the experimental data. And so they look at the data, they start to identify things. They can correlate scores, but they can also choose times to break those scores and say, hey, it's okay to sort of take a bit of a hit on this score because I know we're past the point of the score mattering. And I really believe that this change or these sort of sets of changes are going to have a good impact on the function.
That's like our green points next to red points, next to yellow points. Like it's interesting that like the agent decided to put second heterogeneous, like I guess you said PLDT, and like one cluster. Do you have any like rationale why like see such different PLDTs in the same relative map space, or is it because they're just similar like space or whatever?
Yeah, so I'd say in general, things that are close in the UMAP space are sort of close in sequence and in structure, and so you would expect things that are closer to have like better PLDDT. But I'd say more nuanced. It gets like it's like too high dimensional of a space to really understand. And so like you could have a point here and a point there, and there could be like a huge valley in between them that's on some dimension that's not captured by this, where we couldn't really understand. And so it can give us a sort of vague understanding of the sort of solution spaces that the agents are reasoning over based off of like distances to one another and like relational distance, but not really the it's like not so interpretable in terms of like this means this or that means that, I would say. Oh yeah, should we call it there? I think that's
just try to wrap the next couple minutes.
Yep. Okay. Yeah, I'll cruise through it and have some time at the end for questions, but I'm mostly done here. Yeah.
Almost done. It's a big question. So in this case, it kind of correctly, you're starting with like maybe a solution or a partial solution, you're trying to improve on it. Well, let's say if you're starting with a new problem that there is absolutely no solution. Have you explored agents on that kind of? Let's say you have can't find anything that works.
Mhm.
Like other methods, like could you start with that problem?
I haven't tried it yet, but I have a lot of confidence that it would be able to do it, because I think it can do things like the way that it can use tools. It could just take a sort of tool out of the box, use it to make its own sort of starting solution, or it could do something like condition the fold in a certain way if it wanted to use like a very specific fold to solve the problem. It could do any number of things there, and based off of the sort of tool usability that I've seen, it would be able to find itself a starting point. In this case, they aren't allowed to fine tune, but it's definitely something that you could add in because, like we've seen cases like agents are able to do this sort of thing in terms of model optimization.
I'm curious to ask you a question because you're running a business. So who's going to capture this value now? Are all these capabilities going to be monetized, or where do you see the value of your business coming from the next 5 to 10?
Yeah, I mean, it's a great idea or a great question. And sort of, I think it also is a bigger question for the field as a whole is like, where are we moving towards if we can basically automate the whole process of protein design? In this case, I call the title of this like problem formulation as this frontier because we're setting up the problems for the agents. But what I'm quickly seeing is that the agents are also able to formulate the problems. And so it's getting pretty it's getting interesting, and it's hard to know exactly where we're headed with this because for the business at least, we have this problem formulation of a human innovation. We have a structure that we know is able to confer stability to the antibody and allow us to enable oral delivery. And so we formulated the problem in that way and sort of identified a solution space, and then we'll have the agents take over the rest and do the rest of the reasoning with all the data and everything else. But if you wanted to make a new business and do something else and go beyond that, like you could even rely on the agents to come up with that business if you trusted them and do those sorts of things as well.
Now, I have a sort of skepticism regarding agent that initially we use our own creativity to figure out of distribution sample spaces to be good or right. But now it seems like the further this progresses is more like we sacrifice our entire role of influential not to the agents. And I'm not saying this from like an anti-AI perspective or something, but I'm thinking that if there could be a way for the agent to allow us to work with it, and then like rather than it going through some kind of black box to figure things out, if it could like define its own like objective perspective and like communicate to human like what kind of sample space am I doing, what kind of like structure am I trying to like create, what kind of pipeline so that we can work with the agents rather than like let it get it like sacrifice all of the onto it.
Mm-hmm.
Yeah, because like a lot of like UGP models, they're also allowing us to like interfere with their thinking process, right?
Right.
And I'm thinking that this could be a much better like way to work with an agent rather than just like let it to that.
Yeah. Yeah. So I agree. I think especially because like we as humans still want to formulate the problems that we solve and have some input into the way that we solve them. This is a part that I'm building into this system as well, where the agents can come up with solutions and propose them, and then you can have this sort of reinforcement learning with human feedback, where you say, "I like this solution because of X, Y, Z. I don't like what you've done here. Refine this," and then that becomes more context for the process of the agents. Yeah, I'll wrap up quickly, and then so that we can just have questions at the end, I guess. Yeah, so last little bits here. So a lot of people to thank from the Baker Lab and my PhD work there. And then lastly, I want to thank the team at Optimino as well, which is small but mighty. So Julie, my co-founder and CEO, who's in the back. Brian, our one scientist. Now the team is growing pretty rapidly. Basil, who's a mentor for the company. And then I also want to say that we are hiring ML folks now to expand the team, particularly people with protein design and Asian experience. So if this is you or this is somebody that you know, I would love to chat. And cool, so I'll leave it for any questions as well.
All right, I know we had a lot of questions along the way. This was super fun talk. So please, we've got a few more beers, maybe a little bit of pizza, so stick around. Ask some more questions, Isaac, mingle, and we'll see you next month over on the other side of the river in Cambridge. So yeah, thanks again.